So thank you very much for this opportunity. I really appreciate it, and I look forward to share a bit of our journey in establishing this collaborative biotech research at Ashesi University, and in particular to focus on the open DNA collections and how we're using this to solve some of the problems in health-related fields. So I'll explain in just a little bit what open DNA collection is and how can we use it. But before, I thought I'll start with just a little bit discussing about or introducing Ashesi University to you. And so Ashesi University is one of the top private universities in Ghana. We started quite small, about 20 years ago, with about 30 students in 2005, 4 faculty members and 4 administrators. But we started small with a great vision, with a big vision. The vision of Ashesi University is of a flourishing Africa whose leaders make ethical decisions in the best interest of their communities. And we are driven by innovation that will then support the driving the economic and cultural and social growth. The mission is to educate ethical entrepreneur leaders who will actually be able to bring this reality or this vision to reality. And we also are really interested in collaborating with other institutions and to multiply this work on the continent. So as I mentioned, we started quite small, but today we are quite large. So you can see a picture here of where the university is located currently, is at the top of the hill with this incredible view of a community here, and also of the entire Accra city down here. We currently have just a little bit over 1,500 students. Out of these, 1,430 are undergraduate, and we have started a couple of years ago with a very small collaborative master's program. 95% of our graduates start and begin work within 6 months of completion. Another characteristics that we are very proud of is that we have this 1-to-1 ratio of gender parity and students on scholarships, with a lot of our students, about 40% of our students being on full scholarship, and part of that is due to the MasterCard Foundation. We have ranked in the top 10 among Africa's top most impactful universities in the Times Higher Education ranking. And then we have seen lots of students who have seen learning reforms and stronger outcomes through the Ashesi-led partnership. So this has to do a lot with our collaborative education program, which is all over the continent, and with whom we collaborate and exchange information about education in particular, and how to improve the education across the continent. And then we also have an entrepreneurship center where we support a lot of ventures of our students, and we have this quantification to it as to how many households have been receiving access to climate smart products through the Ashesi Climate Innovation Center, and the people who participated in this particular program. Next, it's maybe a little bit of information as kind of what is guiding us from here on. So as we are stepping into a new decade, we are looking at the global challenges that exist, but also the continental challenges. So as we're all aware, we're going to have a significant population growth in Africa. Climate change is going to be a big problem for which we need to develop solutions. The pandemic definitely have pointed out that there is a need for African science and health leaderships. And I think this is where we also are seeing these seminars being and contributing at the spread and the share of knowledge, and how we can all collaborate to improve this specific gap that we have all noticed, specifically during the pandemic. We're also looking at continental free trade agreement, the fourth industrial revolutions, needing for new tools for problem solving. We know that manufacturing in Africa is going to grow, particularly biotechnology. Manufacturing it's going to start growing in the near future. So based on that, and based on our information that we've got through the pandemic, we started looking at how can Ashesi actually help prepare for these gaps and perhaps minimize them and find particular solution. So one of the approaches that we took was to introduce a new program, which is the BSc Bioengineering. Currently, we have engineering undergraduates in electrical engineering, electrical engineering, mechanical engineering, and computer engineering. We also have computer science and business administration. We also, as I mentioned, we have a little bit of graduate school studies in mechatronics engineering, and now we're adding biological engineering, economics as well, law and public health, and also we're looking at intelligent computing as a graduate program. Particularly the intelligent computing systems and mechatronics will have a ripple effect or an effect into this biotechnology development that we're looking at, and also with the biological engineering specifically, at what areas can we actually make an impact. And so bioengineering is a very large field and has a lot of different areas that you can focus on. We took as our goals to look at what impact we can make in digital health, synthetic and computational biology, and neural engineering. Those three areas are kind of our core areas of interest that we hope to implement into our biological engineering program. So with that in mind, I wanted to focus just a little bit more on what is synthetic biology and why this might be a good area to start with. So synthetic biology is basically the approach of using bacteria or other organisms and engineering them and designing them to produce things that are useful for us or that we would need. And it provides an opportunity for Africa to excel in this area, as it is currently growing globally. So it's an area where if Africa as a continent can start getting involved, we become stakeholders in this development, and not only the receivers of new technology, but we can actually create and invent new technology specifically for our context. It has wide range of applications that are, they're going from energy and agriculture to healthcare to biosensory integration in IoT, so Internet of Things. So you can look at how do you integrate biosensors in monitoring different things. It's also based on the biological software revolution, which is part of the fourth industrial revolution, where we're looking at manipulating DNA and using it as an information storage, for instance, or as a production line for different proteins. And that will also have a big impact. And it's also a real potential to develop a program from the ground up. Education in primary school communities and evolving industries, because this particular trend or line of education is not really existent or developed well in Africa. And so this will provide us the opportunity to start really at the early ages and start introducing and cultivating a spirit of curiosity in the students and then involving communities to help us solve some of these very incredibly large or complex problems that we are facing with. And also tapping into the industrial support to develop this area. It's also something that, if we're switching now to look a little bit more specific at biotechnology, it's a really quite large market. So it's been estimated that in 2020 that the African biotechnology market is about $60 billion, and it will have a very fast annual growth rate of about 9.8% per year. So what is OpenDNA? And so OpenDNA, you can think of it as being the toolbox that allows us to create or develop or engineering two different organisms. So we need certain tools in order to be able to make an organism do what we want to do. And so the idea of an open science is to create this repository or these sets of tools that are available to everyone. They're not under protection of patents, and we are openly sharing all the knowledge that we're finding and we're discovering in order for the progress of the community and the society. And so we are part of this network called ReClone, which stands for Reagent Collaboration Network. And the idea of this Reagent Collaboration Network was to develop community of researchers and scientists that can collaborate. And then they would be able to develop a repository of, for instance, parts, as we were going to be looking at, or activities and informations and research that would be openly available to everyone. As part of this network, we are trying to establish regional reagent hubs. So one of the big problems with the biotechnology is the supply chain, which I'm sure all of you have experienced at some particular point, and not just in science, but I think in science is a little bit more accentuated. Where the supply chain is very sparse, leading to really delays in availability of reagents, particularly if we're looking for diagnostics. We're being, research is being put on hold, or it's being delayed for a significant amount of times because these reagents are not available. We've seen some of this also through the COVID pandemic, right? That has gone even more accentuated, and not being able to have access to vaccines in times and the other medications in time. And then we're also looking, as I said, to build this globally distributed community of researchers to work collaboratively to establish and develop the biotechnology sector. We're right at the beginning, and if we don't work together, we can't really make a big impact into this direction. So we're hoping that with this network, we can create and instill this open collaborations between the scientists. So Ashesi University is one of these regional reagent hubs. So we are holding this collection of open DNA parts. So kind of what are they, and what are we gonna, what can you have access to? So here I'm showing you the specific collections that we currently have. And as a community, we're working on developing new collections. We're working on developing or characterizing these collections so researchers have more information about them and wouldn't have to spend time to do characterizations of them before they can start using them for different applications and different innovations. So this collection is going to grow, but we basically have all of these collections on site, not only here. So we are the regional center for African continent, but there is a regional center for South America, and there's a regional center for Asia. So in the global South, we have at least 3 currently, and we're hoping to grow in the near future, where you would be a lot more easier for people to have access to these collections and be able to develop new innovative approaches to solving problems. So for today, I just wanted to show you kind of one example of what can we do, or what could you do with some of these collections. And specifically, I'm going to look at the open enzyme collections. And so the open enzyme collection is basically pieces of DNA that have been designed by Open Bioeconomy Labs in the UK, which is one of our collaborators as well. and many other collaborators. So people have worked on designing these DNA pieces. So DNA is the recipe for producing proteins. So proteins are things that biological systems use to build things, right? So if we want to build a cell, we need to create specific proteins that we could build the structure of that cells. They were synthesized initially by free genes, and now we receive them also through this through our collaborations through Reclone. They're distributed after under an open MTA. So what that means is that it's a material transfer agreement, meaning that we are willing to provide these at no cost to you, just the shipping that we're looking to have you pay for. And then you can use these particular collections to do any innovations that you might want, either for commercialization purposes or for nonprofit. So you're not bound, we're not bound by any patent restrictions for these parts. Now, as I said, perhaps we can look a little bit in more details. What do we mean by these parts? And so one way to think about it is that a protein, in order to be produced by a cell, requires certain units. So you can think of it as Lego units, right? In order for me to get a Lego car, I need to be able to put the Lego parts to be able to build that car. So similarly, in order for me to produce a protein or an enzyme that I'm looking for, I need to be able to put the correct parts in order to be able to produce it. Now, there's different ways in which you can kind of quote unquote biohack the cell or convince the cell to produce these. And so one of those parts, for instance, is a promoter. You can think of a promoter as a switch. Now you can have a switch that you manually turn on, or you can have a switch that is always on, or you can have a switch that you can turn on a particular signal. So, there's a particular biochemical that it's present in the environment, and that will trigger the production of this particular protein. So that's what the promoter is. A tag, the tag that you see in purple, that's more or less a way for us to be able to purify, to pull that particular protein out of the mixture of the protein that exists in that cell. Then we can look at the reporter, which is a way to tell us if the machinery is working correctly. And that can be a colorimetric, that can be a biochemical signal, depending on what is your interest or your assay it's requiring. You can look at, we could put in a cleavage site where we can cut. So cleavage site is a specific location where you can have biochemical scissors or molecular scissors to actually come and cut things for you. And that depends on your applications. You might need to have this module, or you might not want to have this module. Then we have the actual protein that we're looking for. So the catalytic domain that we'll be looking. So this is the interest of your protein, the interest protein that you're looking for. And then you need to have a switch to turn it off. Just like we had a switch to turn it on, we need to have a switch to turn it off. And so the open enzyme collection gives you access to 6 different promoters, for instance, 8 affinity tags, 2 reporters, 4 cleavage sites, 3 terminators, and 20 coding sequences. And so basically what you have now, you have what we call, refer to it as a sandbox, an area where you can literally play, just like in a sandbox, to design different architectures or different conformations of these proteins to do any specific application that you are interested in. And so if we can look at one particular one, for instance, the protein that has been used in order to do a COVID diagnostic test. So I'm sure at this point, everybody's familiar with the COVID test, a PCR test in particular. And so we have been looking at how to develop one of the enzyme that is critical for the PCR test, which is what we call open vent. It's a thermostable enzyme that's actually able to copy the DNA and amplify, increase the signal that you're getting from this particular DNA for you to be able to determine if there is COVID present in the sample or not. So in order to do that, you can create, so we have this open vent gene in our collection of proteins. You can create it and put it in whatever arrangements you want, and that is referred to as the plasmid. So it's the backbone where you introduce all the pieces. So you can think of it again of the Lego analogy. You have the platform to where you put all the Lego pieces to stay together. That's what your plasmid would be, is that backbone where you can assemble all the parts together. And then you put this particular circuit or this assembly in a particular organism. And that organism can vary, from what you have available or something that we know that has been well characterized. And so we know exactly how it behaves and how it produces our parts. Then we grow that particular protein. Now, we chose, for instance, to have a gene that was under a switch that we could turn on when we decided it to turn it on. So we had this IPT gene induction, which means I'm just basically giving a biochemical to turn the protein production on. And then as the cells are growing, they're making these proteins in abundance. And then I can do some downstream analysis to show that I have this particular protein. In this particular case with the open vent, there's really not necessarily to do any of these further down characterization unless it's important for your application. Otherwise, you can just take the cells and stick them in, mix them with the sample that you're trying to ascertain if it has COVID DNA, for instance, and then you would look to see if that is present in your sample. So in order for us to know that this particular process up here from 1 through 4 actually worked, we also tagged the protein. We put a colorimetric tag to it. So if the bacteria actually makes this protein, it will turn a specific color. And in our case, we turned purple. So you can see here that we have a culture of bacteria that actually have been growing and they're producing this particular protein, the open vent, that we could use then in further down for testing of, let's say, presence of COVID. Now, to make this even more available, I would say, to a low resource setting, we even work to help a little bit and characterize how do we send cells, bacterial cells, to people who don't have access to -80 refrigerations, which is low temperature, which are also quite expensive and bulky, and some constrained resources. You might not have access to those. So what we did, we took the cells and we actually dried them up and kept them on the benchtop. And we have seen that after 50 days, you can still have a lot of growth. So it's a little bit difficult to see, but all the little different dots that you have here is a colony forming unit. So it's a colony of bacteria. They probably have about, hundreds of thousands, if not maybe billions of bacteria that give generation to this colony forming unit that you see. You see a little bit better up here, which, it's a similar thing of bacterial growth, just a different case. This is when we put this construct in the cell. So you can see that there's actually very little difference between having the cells being dried down and rehydrated in order for them to grow, and even if they were not dried down and they were just delivered through a low temperature or control low temperature shipping, for instance. And then the question is, what else can we do with it, right? So here's the diagnostic of the COVID. For instance, we can look at this then we can take them and create different assays, for instance. So I'm sure most of you are familiar with a one-use malaria test. That malaria test is called a lateral flow. We can have different variations of that lateral flow assays by using different microfluidics, which is basically different channels that we can separate things, we can mix things, so we can have multiple assays on a one cassette, similar to the lateral flow. We can use them for education. You can use these proteins for research. You can use them just to share resources with other people who don't have access to them, and allow them now to have this ability to create or innovate new diagnostics or new approaches to develop new drug therapies or things of these nature. I wanted to kind of give you two other applications, for instance, of how we are considering using it. This is based on my background in cancer, where we can introduce these bacteria, we can engineer these bacteria to turn on a gene that produces a drug, a protein-based drug, only in the presence of low oxygen. That low oxygen environment is only specific for tumors. And so you can engineer these bacteria and introduce them in the bloodstream. You can use bacteria, obviously, that's non-pathogenic, that will not create any other diseases. And these bacteria, some of them will be destroyed by your immune system, but some of them will end up in the tumor, as they will end up in perhaps other tissues. But in the other tissues, they cannot, the production of the drug cannot be activated because it's not a low oxygen environment. Only in the tumor, you have this low oxygen environment. So when the bacteria find themselves in this environment, then the gene that's specific for producing the drug will be turned on and will start, coming out, be excreted out from the bacteria into the tumor, leading to the death of the tumor cells. An additional benefit of using bacteria, for instance, rather than using some nanoparticles or some vesicles for delivering these particular therapies, would be is the fact that bacteria tend to stimulate the immune response because they're a foreign object, right? A foreign entity in the body. Tumor cells have developed a very subtle way to escape the immune system. So they tend to downregulate particular receptors, so the immune system can't really detect them. And by introducing these bacteria, what we are hoping, we don't know that, but we're hoping that we will be able to really reactivate and awake these dormant immune cells, that then now, because they are in this tumor microenvironment where there's a lot of tumor cells, then they can start realizing that these tumor cells shouldn't be there and lead to their destruction. Another example, for instance, is for colon cancer. So Cologuard is a new test on the US market that is looking at identifying the early stages of colon cancer. And it does that by taking samples of the stool and identifying specific cells or blood present in the stool. It's not as good as a colonoscopy. Doctors don't like it too much because it misses a lot of the precancerous polyp characteristics. Also has a really high false positive rate, about 13%, which is quite high. So the idea is that now we can increase the specificity, for instance, of one such test by making bacteria present specific receptors that are specific for cancer cells. So we can engineer the bacteria the same way as we had before. Now instead of producing a drug, they will produce a sort of a receptor, or if you think about it, of an antenna, right? That's able to capture either particular cells or specific DNA sequences that are indicative of the presence of these cells. And these constructs are usually have a very high specificity rate. And so we would hope that this approach would lead to a much increased accuracy and specificity in detection of cancer cells or precancerous cells or blood in the stool, or things like that. So it's another way to use these approaches for early detection of cancer. And then, as I mentioned before, what would be interesting is to try to use some of these collections to be able to create new tests. So what I'm showing you here is, rather than that lateral flow test, we are looking at a microfluidics, which basically has, here is a reagent reservoir. So you can bring in particulars like your samples or reagents that you can put here. You can add your bacteria, your samples here, and they will end up mixing, for instance, in this particular chamber, and then they can give you some sort of signal that you can detect in this chamber. This architecture can be easily adapted for whatever application you need to, and also they're fairly easy to produce. And a fairly large scale if you need to. So you can use laser cutters to actually produce these microfluidics. And if you're familiar with microfluidics production, is that usually all these channels are produced by etching, and those require special environments and capabilities to or access to specialized equipment. And so it's not very easy to do. They're also not very easy to modify if you wanted a different architecture. So by using laser cutting, it's a lot more easier to do this. And so Ashesi University has become a part of this global frugal diagnostic network, where we're supporting and trying to stimulate the innovation of diagnostic development in the low resource limited settings. And we have held different online meetings where people have presented and gave different approaches to creating some of these microfluidics or other different approaches to create diagnostics. And then in December last year, we held a hands-on workshop on our campus, where people from all over Africa came and learned how to do microfluidics and also how to do lateral flow assays. And we discussed within the network how to exchange resource availabilities. So if somebody's interested in producing some of these but doesn't have access to some of the instrumentations, we know who to call and who to appeal to be able to start working. And we're also looking at creating, we worked on creating teams that can work together and collaborate together and apply for fundings to be able to produce different diagnostics that are needed on the continent, which for diseases that are only specific to us, and they're not of interest to the global north. So there's no diagnostic for some of those diseases that our physicians encounter on a regular basis. And so this network is trying to really stimulate and support the development of such diagnostic approaches. And so kind of take-home messages here would be that, we're looking for innovative biotech solutions. So I'm hoping that I've been able to, convince you that open science designs are innovative, that there are ways in which you can create new constructs, and you have these sandbox where you can play and develop all sorts of different architectures and structures. That, Ashesi has been, it's making an impact on the healthcare, perhaps not necessarily systems, but the healthcare sector, I would say, by being part of these two particular networks or establishing these two particular networks. And we're hoping that more people who are interested will join us in these networks. And then, that we are the regional distribution of centers for open DNA collections. And so if you are interested, please contact us and let us know what you'd like, and we can help you not only just ship it to you, but we can hopefully be able to support you in establishing whatever protocol you might need if you require that support. If you don't and you already have everything, but all you need is the DNA, would be more than happy to supply those DNA for you. And then you can start producing your locally enzymes for teaching, for research, for diagnostics, for any other innovations that you are working on developing. And I just wanted to leave you with kind of one approach, and not one quote or example, maybe it's more correctly said, of how I envision Ashesi's role, but also I think what everybody on this continent is kind of in a similar situation. There's this railway, simmering railway, which is really a marvel of engineering, and it was developed in harmony with the nature. So it was built as to minimize its footprint in the nature, and so it was built very high in a very mountainous area without destroying that area. But it was built before there was a train capable of completing this steep journey, and the builders had perhaps the faith, but also perhaps the desire and the conviction that there will be a train someday that will be able to make this journey. And so that's how we're looking at our journey into this biotechnology sector on the continent, as we're looking at creating an education incubator, a collaborative incubator for creating, engineers and innovators that can undertake and develop this biotechnology industry in Ghana, but not only in Ghana and anywhere else on the continent. So thank you very much for your attention, and I am ready to take all your questions.